Good morning, Philippines! I am Eloisa Cruz, your news anchor, and this is Biology News Today.
This is Channels Television's breaking news.
After nearly 3 decades in prison for a crime he didn't commit, Richard Toller was exonerated in May 2010. For years, Toller and the Ohio Innocence Project sought DNA testing in the case, and the fourth round of testing finally proved his innocence. An Ohio judge choked back as she ordered his release on May 5, 2010. On May 24, 1981, an 11-year-old girl and her 12-year-old male cousin were walking their bikes in Cleveland. The man pulled the gun and assaulted the boy, forcing him to lie on the ground, while he raped and sexually assaulted the young girl. The only physical evidence presented at Toller's trial came from a forensic analyst who had microscopically examined a hair comb from the victim. Despite Toller's alibi and the lack of physical evidence tying him to the crime, he was convicted and sentenced to life, the sentence required by law because the victim was less than 13 years old. An additional 12 to 40 years were added to his sentence for the crimes against the boy. More than 20 years after Toller was convicted, Ohio enacted a law allowing DNA testing in very limited circumstances, finally clearing him up more than 28 years in prison. Days later, Toller was a free man. He was 24 years old when he was convicted and 52 on the day of his release. A big thanks to the DNA testing. So today, we will know more about DNA technology that helps Mr. Raymond Toller to prove his innocence. We will ask Kyla Kulis for further information. Kyla, what is DNA technology?
Good day, viewers! In today's news, different information regarding DNA technology will be proliferated. Deoxyribonucleic acid, or known as DNA, is an organism's genetic material inherited from one generation to the next. It is said that it holds many clues that have unlocked some of the mysteries behind human behavior, disease, evolution, and aging. According to Khan Academy, DNA technology is the sequencing, analysis, and cutting and pasting of DNA.
So Kyla, what are the forms of DNA?
Its common forms include DNA sequencing, polymerase chain reaction, DNA cloning, and gel electrophoresis.
What are the advantages of DNA-based technology? Where can we use it?
Moreover, as technological advances lead to a better understanding of DNA, new DNA-based technologies will emerge. Recent advances in DNA technology include cloning, PCR, recombinant DNA technology, DNA fingerprinting, gene therapy, DNA microarray technology, and DNA profiling. These DNA-based technologies have already begun to shape medicine, forensic sciences, environmental sciences, and national security. Indeed, DNA technology is very essential in the present days. It makes a certain process more convenient, especially in terms of genetic engineering. This This is Kyla Kulis reporting.
Thank you, Miss Carla Coulis. And now we will ask Rachel Robles, purposes of DNA technology and how does it works?
DNA technology is the study and manipulation of genetic material, and scientists are using DNA technology for a wide variety of purposes and products. A major component of DNA is what we call the cloning, which is the process of making multiple and identical copies of a gene. There are 5 steps in cloning. First one is extract DNA from a donor. Second, prepare an egg cell. Third, insert somatic cell material. Fourth, convince the egg that is fertilized and implant it. And last, repeat until viability. Another thing, DNA technology has also a major impact on pharmaceutical industry, agriculture, disease therapy, and even in the crime scene investigations. So, let's take a closer look at the application of such an important field of the study. Pharmaceuticals and medicine. DNA technology is being used to help diagnose genetic diseases such as sickle cell and Huntington's disease. Since these diseases are transferred genetically from one generation to the next, those who have such diseases can be identified, sometimes even before birth, and be treated before symptoms appear. Next is the vaccines. DNA technology is also used to make vaccines such as altering the pathogen's genes and mimicking surface proteins of harmful pathogens. Those vaccines from DNA technology will help you trick your body into fighting harmless version of pathogen so that if you are exposed to a harmful pathogen, you have already built up defenses. Last one is the agriculture. DNA technology doesn't just make bigger and better food though. It's also being used to create food products that have medical benefits. Example, a new rice strain has been engineered that has milk proteins that can be used to treat infant diarrhea, a corn that can treat cystic fibrosis, and a duckweed to treat hepatitis. Those are the purpose and uses of DNA technology. This is Rachel Ann Robles reporting.
Thank you, Miss Rachel. We will be back after a quick commercial. The secret behind this new DNA technology and how is it being used in identifying the real culprit? Revealed. Let's ask Rosalinda Cueto for a report.
Good day, viewers. This is Rosalinda Cueto for Biology News. DNA technology is a field that has many contributions in our lives, and one of its most important contributions is in the industry of criminal investigation. Before DNA, criminal justices and investigators just relied on things like bloody footprints and hair color, but 1986 changed all of that when English geneticist Alex Jeffrey developed the first ever application of DNA science to be used in a criminal investigation, the DNA fingerprinting. It allows DNA samples from different people to be compared to look for similarities and differences. The big question now is, how is it being used, or is there a process for it to work?
Any two people in the world have 99.9% of their DNA the same, so this process analyzes the differences on the remaining 0.1%. It is a very sensitive technique which only need a few skin cells, a hair root, or a tiny amount of blood or saliva. A cell sample is collected by forensic scientists and investigate it in the lab. First, they extract DNA from the cells. The copies of this DNA may be made by polymerase chain reaction or PCR. Then they use enzymes to cut the DNA into shorter fragments. The fragments are then placed on a special well called agarose for the process of gel electrophoresis. And finally, the lines produced by the DNA sample from different people are compared. They look for the one that is most similar to the one from the crime scene. Let us use this example. An item was stolen in a shop. A drop of blood was left behind by the thief. Samples of DNA was taken from 4 suspects and compared to the sample left at the crime scene. Which suspect is guilty? Here, suspect 3 is guilty. Why? This is because the patterns of bands of suspect 3 really matches the bands from the crime scene. Their DNA are the same.
And that's it for my today's news. Back to you, Elon.
Thank you, Rosalinda, for that report. And now Ms. Rayan Ronquillo is currently online to explain the practical applications of DNA technology.
Practical uses of DNA technology. Throughout the billions of years in life history, it brought a lot of changes. DNA technology enlightens us about our life history. It also prevents and eases of human suffering. It protects our environment. And lastly, it enforces our law. In the line of medicine, it was considered as the most familiar application of DNA technology. For an instance, technician can make an advanced test to the person's DNA for many alleles associated with an inherited disease. Disease screening and diagnosis can be an advanced move of a technician into that person. Moreover, according to Marielle Hofnagel, who is a professor in Department of Biology, if genes can be used to cure cancer, it must also be possible to use DNA to make a healthy person even better. She also said that it should be possible to inject genes that make an athlete stronger, faster, or better able to withstand the physical stress of competition. Gene doping is the use of DNA to enhance the function of a healthy person. Another application of DNA technology is genetically modified organisms, or GMOs. It finds a way into the human food supply. It has a great help in agriculture. Even though many microbes, plants, and animals have been genetically modified, scientists first accomplished this feat of genetic engineering in E. coli bacteria in 1970. DNA profiling is another use of DNA technology which help to solve crime or match parent with their offspring. In criminal justice system, if an investigator has no leads for the suspect in a crime, DNA profiling will make its way. The database of DNA profiles were compared to the DNA collected from a crime scene. If someone's DNA profile matched to the record file of the investigator, they can be considered as a culprit. Food safety relies as well on DNA testing. Many of us use DNA to know the history of our family. The only thing which can verify that a man is the father of a child is through DNA testing. Back to you, Eloy.
So today we discovered and learned what's DNA technology, its definition, purpose, how does it work, and even know the practical application of it. So that's it for Biology News today.